Hello, everyone. Welcome to the Sci 113 Science and Society Speaker Series. I'm really excited to introduce our guest scientists for today, but just for a few reminders for those of you joining live, thanks for joining us. I hope you all had a bit of a relaxing few days off for the midterm break. And I know you're all working hard on your term paper projects for Sci 113. As you're watching the recording or during the seminar live, definitely think about some questions that you would like to ask today's guest speaker. There will be time at the end of today's talk to answer your great questions. There's been such great questions over the course of the speaker series this term. So well done to all of you, and we'll get a chance to address all your questions then. Just as a reminder for folks to, ensure your questions and comments in the chat are respectful, but definitely feel free to raise your hand as well, and we'll get to those questions. With that, it's my absolute pleasure to introduce today's speaker, Dr. Sally Otto. Dr. Otto is a professor in the Department of Zoology and a researcher at the Biodiversity Research Center here at UBC. Dr. Otto is a world-renowned scientist and has received numerous awards, including the coveted MacArthur Fellowship, popularly known as the Genius Award, and was recently elected to the Fellowship of the United Kingdom's Royal Society. In her research, Dr. Otto explores really fundamental questions about evolution and how and why we see diversity among several different types of biological traits in the natural world. Dr. Otto's work addresses these questions using a combination of approaches, including mathematical modeling, statistics, and even evolutionary experiments. So thank you very much for joining us, Dr. Otto, and I'm really looking forward to your talk. Thanks for joining us.
Thank you. Can you hear me?
Yeah, sure can.
So I was asked to give a little bit about my journey to where I am now as a scientist. And I think when I was at your stage, first year in university, I really had loved all my courses in biology, and I'd loved all of my courses in math. I'd read some of Darwin's work, I'd read some of Mendel's work, but I wanted to join math and biology in some way. And I thought, aha, I'm going to be a genetic engineer, because genetic engineers there must be math in genetic engineering. Well, I got to university and I did the equivalent of the work-learn program, and I was in a lab, a genetic engineering lab. And I found out that there was no math in genetic engineering, at least not most of the time. It was mainly molecular biology and a lot of pipetting, at that time, radioactive invisible liquids. And it was not for me. It was not what I enjoyed doing. But I had a wonderful mentor, and my mentor talked with me about my interests and guided me to talk with another professor. And just her name was, the first professor was Dr. Virginia Walbut. And we went on to work on a paper together, marrying math and biology. But she sent me to talk to her colleague, and her colleague said, I have a project that might need a model. And he told me about this system. So this is my new advisor, Mark Feldman. He told me about this system with 3 sexes. It was a clam shrimp, Eulimnatia texana, shown here. And there were 3 sexes genotypically, 2 types of hermaphrodite that made both eggs and sperm, and males. 3 types of sexes, that's weird. And what it begged the question, well, why doesn't it just drop the males and become a selfing hermaphroditic population? Or what keeps the system with 2 types of females rather than just 1? And so with those kind of puzzles in hand and talking with our colleague Clay Sassaman, who was working with this shrimp, we built a model. And that eventually led to my honors thesis and a model of this clam shrimp, Eulimnatia texana. But I have to say I was hooked, because the idea of being able to take these mysteries and make sense of them through equations was just I took to it like a fish to water. It helped from those models. We were able to say things like, well, what would the fraction of males be at steady state? And turning that around, what do those equilibrium fractions tell us about the underlying processes at work? Inbreeding depression, mating interactions. So we could infer those from the answers that Clay was getting in empirical work. So that was the start of my career. I went on and did a PhD at Stanford with Mark Feldman, and then went on and postdoc at Berkeley first, and then Edinburgh. And in 1995, I joined the faculty here at UBC, and I have really enjoyed my colleagues and my work, my students, and my interactions with everyone here at UBC. So today's lecture is going to give a bit of an overview of what evolution is from a mathematical perspective. So I'm bringing this bridge between the evolution as a process and the math fundamentals behind it. And then I'm going to talk to you about how that understanding of evolution has shaped science and policy today. So let's start at the beginning. The first part is really a kind of evolution 101, the core fundamentals of evolution, and then we'll talk about application. So just to be clear, evolution simply means change. And in biology, we refer to the change in form, morphology, the physiology, the cell biology, or the behavior of organisms over time, over generations. And this is what Darwin called descent with modification. Now, because evolution just means change, change can occur for any number of reasons, and that can be chance events. Some types get hit by lightning, others don't. Some have a bonanza number of kids because they just found great resources, others didn't. So that's chance bursts of reproduction or death. Mutation causes change, as does natural selection. But it is this last process, natural selection, that explains how kind of these undirected changes, these random events and mutations, lead to an improved match between an organism and its environment. So to explain camouflage, to explain how organisms have adapted the depths of hydrothermal vents to the most desert-like environments on the planet, we need a process that explains how that match comes to be. And that process is evolution by natural selection. So this is a process whereby some individuals contribute more offspring to the next generation as a consequence of their carrying a trait or traits favorable to survival and or reproduction. So in these sunflowers, for example, the first seeds to arrive on that dune may not have been, sand dune may not have been that adapted to the sand dune. But some of those seeds may have been just a little bit more likely to tolerate the drought conditions or grow rapidly down in their roots to reach the water table, and so be better able to survive and reproduce. So just to recap, evolution is change of any sort. Evolution by natural selection is the process that we need to explain the match of organisms to their environment. So evolution has a logical structure that I'm going to talk about in this slide. So individuals must vary in some trait. There must be variance in a trait. And that's patently obvious if we look around in any species that there is variability. In addition, individuals with some trait values must be more likely to live and or reproduce. And that's not always going to be the case, but for some traits, like the speed with which a sunflower may set roots, are more likely to live and or reproduce, and that's selection. And then finally, for there to be evolutionary change by natural selection, those parents that survive and reproduce have to have offspring that are more similar to them, that have traits like fast root development. So there has to be what we say is heritability, similarity in the offspring, based on their parents' values. And of course, when Darwin first came to the idea of evolution by natural selection, he didn't understand that heritability was due to the inheritance of genes, but we now know that to be true. But as long as you have these three features, variance, selective differences, and heritability, then it must be the case that the offspring will carry a different set of traits than the parental generation, because those offspring represent preferentially those parents that varied and carried variants that made them more likely to survive and reproduce. And then those offspring also were more likely to carry those traits. So with these three premises, evolution by natural selection must occur. So that's at a kind of logical level. That those same processes can be made mathematical. And by the 1930s, the core mathematical structure underlying evolutionary change had been described. And I'm going to walk us through the simplest of those models, this description of evolution. And I'm going to keep it simple by modeling only one genetic site within the genome. And I'm only going to take a look at two variants, and I'm going to call them big A and little a. Geneticists call these variants alleles. And we're going to keep it very simple and think about a haploid organism. A haploid organism is one that has only one copy of each gene, like this Eulothrix green algae or bacteria or viruses. Or moss, they carry one copy of each gene. We are diploid. We carry two copies of each gene, so I'm not going to get to the complexity of evolution in diploids. So to model selection or evolution by natural selection, we have to have these selective differences between individuals. And I'm just going to use this symbol, W, to denote fitness. So that's fitness is the average survival and reproductive capacity of a type, either big A for the big individuals carrying that allele, or little a for the individuals carrying the alternative allele. So here's the core of the mathematics in simplified form. We have two types, big A and little a. And at their initial frequency, at the beginning of the generation, I'm just going to call that P. If there's a frequency P of big A allele, the rest of the population, Q or 1 minus P, carry the little a allele. So this describes the whole population at the beginning. Then selection happens. Some of them are big A's are maybe more likely to survive or more likely to give birth, but that's captured by its fitness. So these frequencies change following selection to be in proportion W big A times P in relative to W little a times Q. So for example, if none of the little a alleles survive or reproduce, W little a would be 0, and all of a sudden the whole population would go to being big A. If we have these relative frequencies after selection, those relative frequencies don't sum to 1. And so we convert these ratios of big A to little a into the frequency of big A by just taking the amount of big A alleles we have after selection and dividing it by the total population that we have after selection, which is the denominator here. So this equation maps or takes us through one generation of selection from a starting allele frequency of big A at some time t to its frequency in the next generation at time t plus 1. So from one generation to the next. We can also ask how much change was actually accomplished by evolution by natural selection. That is, how much did P change from one generation to the next by just subtracting off its previous value. This delta here is often used to denote a change in science. So this is a change in allele frequency from time t plus 1, from time t to the next generation time t plus 1. And with a little bit of algebra, you can show that you take this, you subtract off P, and you get this form. And so that's kind of the fundamental underpinnings or workings of evolution by natural selection. But we can actually get it to match the logical structure of evolution by natural selection by going through these three premises one by one and mapping it to that equation. So first of all, you have to have individuals that vary. And that's captured by this P times Q term. Remember, P was the frequency of big A alleles, Q was the frequency of little a alleles, and you have to have both around in order for evolution by natural selection to work. If P were 1 and everybody carried big A alleles, there'd be no change because Q would be 0 and there'd be no change. If Q was 1 and P was 0, same, no change. And that leads to this classic evolutionary dynamical equation where over time, this is the frequency of one of the alleles here, big A, it always follows this S-shaped curve. There's very little change when big A is rare, very little change when big A is common. And that's because there's very little genetic variance in the population. Most of the individuals are little a, or most of the individuals are big A. And there's just not much for natural selection, not much variance to work on. Evolution happens fastest when we have a lot of variation, a lot of a mixture of big A and little a alleles. So that matches the variance needed by evolution to this part of the equation. The next part of the equation reflects selection. There has to be trait differences leading to fitness differences. In this particular S-shaped curve that I've drawn above there, I've made the big A allele 20% more fit than the little a allele, just by choosing these values for the fitnesses. And so there is selection because they have differences in fitness. If the fitnesses were the same, there would be no evolutionary change by natural selection. If I make that difference higher, and so big A is 30% more fit, I see a faster S-shaped rise in the big A allele. Or I can make it slower. So selection is capturing these fitness differences. Is the big A allele 10% more fit, 30% more fit, or 20% more fit than the little a allele? And finally, the third requirement we have to have for evolution by natural selection is heritability. And that's kind of implicit in this model because I said that P was the frequency of the big A allele, and I didn't talk about mutations or anything else going on. So big A parent gives rise to a big A offspring, and there's heritability in this system. And with all of those three premises in place, we will see evolutionary change. This delta P will be non-zero. And alleles that improve fitness, improve the match to the environment, like those sunflowers with faster growing roots, spread over time. So in my lab, mainly we use a lot of mathematical tools and techniques to ask not just how does evolution proceed at one locus, but how does it proceed to explain the diversity of life on this planet with many different life cycles, many different genome structures, ways of reproducing. So that's the kind of core, and I won't get into that today, but I'm happy to take questions about it. In addition, we work in the lab with yeast. Saccharomyces cerevisiae is the budding yeast. Here it is budding. That's a mitotic offspring produced by a parent cell. And Saccharomyces cerevisiae are the workhorses of brewing and bread making. So wine, beer, and bread rely on the Saccharomyces cerevisiae chewing up all of the sugars and turning them into little bubbles and ethanol. But they're also fast growing. Every 90 minutes they divide. And that means that we can watch over the course of a day, evolution happen. And so we will use this evolutionary workhorse to basically ask, how can yeast adapt to a changing environment? And in the lab right now, we're working in the past, we've worked on fungicides and antibiotic resistance, and now we're looking at heavy metal toxins. And what are the limits of evolutionary adaptation? Yes, evolution can lead to organisms being better adapted to the current environment, but there is a limit. There is some place where there's just no mutation that's going to allow survival. And so we're trying to figure out those limits. So within yeast, what we'll find is over the course of days, mutations will arise and fix. We can track the cell density of those adaptive mutations that are able to grow in these harsh conditions. We can sequence those mutations and find out where in the genome they are and what they do. So I won't talk more about that, but that's just another aspect of the work we do in our lab. So in short, evolution happens. It happens everywhere all the time. It may be slow or fast, but it never stops. And nowhere have we known that to be so much the case as in the pandemic that we are still in, the SARS-CoV-2 pandemic. So that's what I want to talk about for the rest of this lecture. What has evolution found out about SARS-CoV-2? So just stepping back to December 2019, when the first reports emerged from Wuhan, China, of a new respiratory illness, within weeks, the RNA sequence underlying the virus was published and shared, and people started searching those genomic sequences for signs of selection, for adaptation to humans from whatever animal host it was in before. And there's still uncertainty. Clearly, there was a bat host somewhere in its evolutionary past, but the most recent host, we do not know. So here is a picture that I'm going to walk us through of some of these genomic sequences. So I grabbed this from March of 2021. There's just been an enormous amount of sequences that have been shared publicly in a database called GASAID. So in this plot, this is a phylogenetic tree, but it's arrayed in a circle. And what does that mean? It shows the relatedness. Every single circle is a sampled virus that was sampled at a particular point in time. So you can see the time axis here. This is 2020 December, 2020 August, April 2020. And right there in the middle is November, December 2019. And so we're saying that people have been sampling viruses from patients all throughout the pandemic. So that's why we have circles all the way along. The closer the circles are, if you have to go down, find their common ancestor and back up, the closer they are, the more similar their genetic sequences are. And that's how this type of tree is made. So from this phylogenetic tree showing the relationships among those sequences and how closely related they are, we can find out a number of things. First of all, SARS-CoV-2 evolved from this common ancestor in late 2019. So it wasn't like it was circulating in the world and had a lot of variation before this. All sequences that we know about share this kind of common ancestor in late 2019. Furthermore, we can scour the genomes and count how many mutational changes are there as a function of time. And that'll give us the mutation rate. How quickly does this virus evolve? And roughly, so this is 0.0008 base pair changes per base pair per year. And across the 30,000 base pairs of its genome, that's about once per genome every 2 weeks. So it's not that fast, but it's also not staying the same. And so we're seeing from this ancestor where there was no genetic variation initially, lots of genetic variants emerging. So as an evolutionary biologist, this is a little bit more like something that evolutionary biologists would see. But I look at this tree and I notice that almost all of the lineages have equal chances of success. So I'm not seeing signs of one type being much more fit than the other. Because if that were the case, I'd see one growing at a much faster rate, one color growing at a much faster rate than other lineages of other colors. I'm not really seeing that in this first year. Most of these lineages are growing at roughly the same rate, and we're seeing a lot of diversity being maintained. So that first year of the pandemic did not show strong signs of selection. Mostly it was this star-shaped phylogeny. But then, as you all know, the first variant of concern was reported by Public Health England in December 2020. This was the variant B117 that came to be known as Alpha. And so before that, we'd seen frequency changes sometimes wobbling up and sometimes down, but this was the first time we saw consistent changes over time and over location, which is indicative of selection, that there was a variant that was selectively favored again and again over time and place. And so if we look here, this is going to show in purple the frequency of the alpha variant, again, spreading over time and over space. So the second year of the pandemic, as you know, and the third year of the pandemic has then been shaped much more strongly by selection, with these characteristic the phylogenies that show a variant that rises and spread really rapidly. And it's hard to see here. If I went in to an earlier circle, you would have seen the alpha wave taking off and really spreading out. And here I'm showing in the second year, the delta wave at one point spreading out and really increasing in frequency globally across the planet. And then more on the outer rings, closer to the present, we see Omicron arising and spreading in the last couple of years. So this is a signature of selection, that Delta was more fit than what came before, that Omicron was more fit than what came before. But also notice it's not like Omicron emerged from within Delta. That'll come back. We'll talk about that maybe later. Omicron emerged from a different lineage. Delta emerged not from Alpha, but also branched deep in the tree. So, and even today, we continue to have a lot of genomic information. I got this from this morning. There's 17 million genomes of this virus that you can download from this database, GISAID. Never before have we had so much information about the evolutionary process as we have for this virus. So I'm going to talk to you about a team that I've been a part of, a Canadian coronavirus variants rapid response network. And we've been in charge of computational biology and modeling of the virus and how it's evolving in Canada. So I'm going to talk to you about some of the material we've developed. Open source databases and open source tools that scientists can use and adapt as they want to track evolutionary change. So this all comes from our website here, and I'm going to be showing you pictures from Duotang, and that's our notebook where we share evolutionary analysis. So we built this automated analysis that scours GISAID, and scours the data that we're getting, and then automates the process of interpreting that data and tracking changes over time and the frequency of that data. I have to be a little careful. I have to say, we don't use GISAID, and the reason we don't use GISAID is it's not, you can't share the data. So we developed as part of an additional team portal, VirusSeq, for all of the Canadian sequence data that we can use and share. So this is all coming from that VirusSeq Canadian portal. So in colors are the different variants, and you can see these kind of wave of alpha and gamma, that earliest wave, and a wave of delta, and then a wave of Omicron, and so on. And the current wave we're in is a wave, it's an Omicron type, but it's BA.2.86. So just to give you a handle on just how much evolution is happening, in the last 4 months alone, there are 257 named lineages of SARS-CoV-2. And here are some of the nicknames. And if it's like alphabet soup to you, it is a little like alphabet soup. These variants continue to evolve. They carry new mutations. And when those mutations are different and that make the lineage different enough, then those lineages get a new name. So these are some of the names of the currently circulating lineages. So like KP3.1.1 was the predominant lineage before the last couple of months, and that's in red. But you can see most recently XEC, that's a recombinant X. Whenever you see an X, that means it's a recombination between two other lineages. In this case, that's a recombination event. between two BA286. So that means two different kinds of this virus got into a single person. They were co-infected with both of them. And rather than just copying one of the genomes, it started on one and then got displaced and finished copying on the other genome. And that's a recombination event in a virus. But in all of these are in the Omicron family. So 257, that's going to be impossible for us to track one by hand. So we need to automate this process. What we do is we estimate selection and the growth in the rate at which each of the variants are spreading to find out which ones are spreading faster and determining why those are spreading faster than others. And we do this both for the main lineages circulating at any one point in time, and then I'll talk later about each variant separately, each of those 257 lineages. But in this graph, we've grouped them all into major lineages. The KP3 lineage, these recombinant XEC lineages, MC, another one in the related family, and all the rest. And these are like the S-shaped curve that I showed you earlier, but now we have all these 4 types rather than just 2 types. And we estimate selection for these selection coefficients for each of these major clades relative to a reference. We choose one of the types as a reference and measure selection relative to that. But how do we do that? Well, let's go back to that model that I talked about earlier. It's based on that very model that we walked through, where you have 2 types, big A and little a. Now we're talking about 2 variants of a virus. And I just showed you how to derive this equation after one time step, but you can generalize that math to tell us what will happen over a period of time t. So this is saying over many days, if we start on day 0 someday, and then go forward in time by t days, this will predict what the allele frequency will be after t days. I'm going to measure selection in a particular way. I'm going to measure selection so it's the log relative fitness of the 2 types. And then that allows me to rewrite this equation in this form. And so that's what we're trying to estimate is what is selection. So we take data from GASAID. Here's an example plot where on these dots represent what the number of sequences on a particular day, the proportion of sequences on a particular day. And early on in April of 2022, they were mainly BA2. The size of the dot is how many sequences there were available. But over time, BA5 started to spread, and you see this blue type rising in frequency and the red type declining in frequency. But on any one day, you have a number of sequences of each type, the red type and the blue type. And I want to know how much is BA5 selectively favored over BA2 given this data. So it's data analysis. How do I do that? Well, on any one day, we observe a number of sequences, like, I don't know, maybe 100 on this day. And there's a certain number of blue and a certain number of red of BA5 and BA2. And so that tells me the chance of that. If I knew what the allele frequency of BA5 was on any particular day, I would expect a binomial probability of observing the data with J observed blues and K observed reds. And I don't know if you've seen the binomial formula before. You might not have yet, but this will be in your statistics class in the future. This is basically saying, what is the chance from a sample of N to observe J blues and K reds given a current allele frequency of P? And that tells us what's the chance of seeing that one day's worth of data. But I multiply the chance of seeing that day's data by the chance of seeing the next day's data, and so on and so forth. And that allows me to estimate selection that will maximize the likelihood of seeing a curve like this one. What must this selection coefficient be in order for P to change, the frequency of the blue allele, in such a way that it best matches the data? There's a second way of estimating selection, and I think it also teaches us a little something. And that is, if I took this equation for the change in allele frequency over time, And I divided the frequency of big A by, there's a similar equation for the frequency of little a, that's Q. And so I work on the ratio of one type of virus relative to the other type of virus. Then that grows exponentially over time, depending on that selection coefficient. Furthermore, if I log it, take the log of both sides and do a little bit of rearrangement of the right-hand side, it turns out that the log of the ratio changes from one time point 0 to some future time point t as a linear function of time, s times t. And that means we can use standard linear regression or other approaches to estimate the selection coefficient as the slope on a curve that has, on a log scale, p over q. These are called logit plots. And if I plot p over q on a log scale, I predict a straight line of its selection. That wouldn't be true if it were a super spreader. If it were a super spreader, I wouldn't expect a line with a positive slope. I would expect the frequency of a type to just jump up at one point in time because there was a concert or some other event that led to a rapid rise in frequency. But if we see the data the data from day to day aligning along a line, that's consistent with selection. It's exactly what this model would predict. So what about today? So I grabbed this from the duotang, the current duotang, and that's as using these tools is exactly, actually we use the likelihood tool, is exactly how we estimate the selection coefficients. So this estimate tells us that at the moment, this larger group of XEC, shown down here in gray, is growing relative to the KP3 group at a rate of 8% per day. And on a logit plot, the relative proportion P over Q to the reference, notice that that is showing relatively straight lines like we predict for selection. And it's really been remarkable as an evolutionary biologist how predictable evolutionary change has been. The alleles are rising. Once they are there and we know what they are, their trajectory is very repeatable and predictable over the course of a few months. The prediction of new variants, that's harder, but the prediction of the spread of current variants is rock solid. So I mentioned, though, that there were 257 of these lineages. I was grouping them before, but we can also look at every single one of them separately. And we do that in this automated package using some reference that's been segregating in the population. So this is all relative to KP3.1.1. And that the leaders on that selection chart are shown here. These are the ones that have the greatest growth advantage in terms of that selection percentage per day. And this allows us over time to find out what's taking off. And in this case, it's XEC, that recombinant I mentioned. You can see here how it's rising over time at the start of that S-shaped curve, and you can predict how it's going to continue to rise over the next couple of months. Now that we've broken it specifically down into this particular variant, XEC shows a 5% selective advantage over the previous lineage that was spreading KP3.1.1. And as I mentioned, XEC is a recombinant, and it carries particular amino acid changes in its spike protein that help it evade immunity. But, it's not just evolution we want to know about. We want to know about cases and what's going to happen to case numbers in the near future. This is actually, this is actually where it's harder to predict what's going on. As I mentioned, evolution is actually surprisingly easy to predict. But what's hard to predict is where the case numbers are going. And that's for a number of reasons. For one, people change behavior. Cases start to rise. People wear masks more often. They go in to get their vaccines. A good time to get your vaccine, by the way, because cases are pretty high at the moment. They change their behavior, or new variants arise, or the other problem is that it's getting harder and harder to get a reliable source of data. So these case numbers depend, the number of cases that we know about depends on how much testing we're doing. And that testing fraction has really dropped recently. These are mainly tests for people that are going into hospital. But over the last 4 months, that's roughly the same kind of rules for detecting cases. So over the long term, a lot of changes in case prevalence reflect who are we testing. But over this month, 4-month period, it's a relatively similar, fairly constant testing regime. So the green dots show the number of detections per 100,000 individuals in Canada. And you can see from this that it's been growing lately. It never disappeared in the summer. And we're not seeing the variants go away in the summer, not like the flu, which really goes away in the summer. But it's more wobbling up and down at a frequency, these ups and downs by a factor of 2, maybe 4, but not going away. So what you can see is recently it started to trend down, and that looks like great news. But look underneath the hood. Underneath the hood, I've put the selection and the lineage changes that we know about under the total number of cases in the green dots. And that's where you see a much different picture. While the overall there's no growth, this XEC variant, that's this whatever color that is, gray, is actually growing, and it's growing at an exponential growth rate of 3% per day. So that allows us to predict, are there any variants that are growing fast enough that we're going to see increased growth over the future? And of course, now we're into November and we are seeing again another uptick in cases, and it's being driven by this XEC variant. So this is very helpful. It allows us to not only get an overall picture of how case numbers are trending at the moment, but what they're likely to do in the near future as some subtypes are evolving and spreading fast enough to cause this curve to turn around. This provides advance warning about what changes to expect in the future and when. And this is a tool that's been used by, we've worked together with the Public Health Agency of Canada, and so it's a tool they use both internally now as well as through Duotang. So just, I'm going to spend another 5 or 10 minutes and then I'll open up to questions. But just, I wanted to share some reflections as an evolutionary biologist working alongside other scientists to help monitor and predict what was going on in this pandemic. There were really 2 big surprises for me as an evolutionary biologist. The first is that SARS-CoV-2 evolved faster than anything we've ever seen. So nothing in the natural world has evolved as fast as SARS-CoV-2 has been evolving. So it's really kind of redefined what a selection coefficient can be in my mind. Here I've plotted, this is just from our reports over the years, what the selection coefficient was for each of the main variants of concern. And these little black arrows are just telling me what the reference was. So this is a selection coefficient for Delta, and it was like 8% more fit than Alpha and Gamma. Alpha and Gamma were 8%, sorry, that's more like 9%. These are like 8% more fit than the Wuhan strain that was beforehand. Omicron was about 25% more fit than the Delta variant that was circulating at the time. And so you can see relative to the previously circulating strains that the selection coefficients are often around 8 to 10% and as high as 25%. It's hard to actually interpret selection coefficients in this way, so I put a little sidebar. Well, first of all, that's selection per day. And on this sidebar, I interpret it. A selection coefficient, let's start at the bottom, of 1% per day means that a type will double in 70 days. For XEC, it had a selection coefficient of 5%. That means it'll double in relative frequency in 2 weeks. But sometimes, like we're seeing selection coefficients more like 10%. And that means weekly doubling. A thing that was rare 1 week doubles in frequency the next week and the week after and week after. And so really quickly comes to dominate. And of course, the one with the strongest selection coefficient, the variant of concern with the strongest selection coefficient was Omicron. Its selection coefficient was 25% per day, astronomical. And that led to doubling times of 2 to 3 days, meaning it went from really not present in a country to dominating the country in a couple of weeks. So because of this continual evolution, this continual appearance of new variants that evade immunity or that get into our cells a little bit better, we continue to see new variants appearing that have a selection coefficient that allows it to spread through, selective advantage that allows it to spread through populations. So variants are continuing to evolve. And when you couple that with waning immunity, our immunity to this virus, unfortunately, doesn't last forever. So if it's been more than 6 months since you last had a case or last had a vaccine, you probably don't have that much immunity in terms of antibodies. So go get that vaccine. You can boost up your antibodies, and that can help protect you against another case. But because that immunity from antibodies wanes, those antibodies disappear after a while, the virus has plenty of individuals to infect, and this drives ongoing waves of infection with a combination of waning immunity and evolution. The other big surprise was that we didn't always see this kind of gradual evolution. Oftentimes we did. So by gradual evolution, I meant this kind of accumulation of mutations over time so that the sequences accumulated became more and more different from their ancestor. This is a mutations over time plot. On this axis is the number of mutational differences in the genome sequence. This is time, and each dot is a sample. So if you focus on the gray dots for a minute, that's the original Wuhan strain, and you can see that they're from their common ancestor in late 2019, gradually accumulating mutations along this slope. And so mutating along over time, they're each a little different from their ancestor, blah, 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 blah. But then new variants appeared, and those new variants tended to have the same slope. So their mutation rate was roughly the same. Their processes of mutation were roughly the same. But they often had a jump up in the number of mutations they carried. So these variants of concern would appear, but they would appear out of nowhere, and they would appear to have more mutations, like there was a jump or a saltation event. We saw that with Alpha. That had, I think, about 9 more mutations. We saw it with Omicron that had about 20 more mutations. We saw it recently with BA2.86, which had 40 more mutations than the other circulating strains at the time. So this is weird. What created this pulse of mutations? And that's not the only weird thing about these variants of concern. The other weird thing about the variant of concern is actually it was pretty different when we first detected it. And that was true for Alpha as well. It was already pretty different. We saw kind of all of these mutational differences, but where was it? Same with Omicron. Here I'm showing the evolutionary tree of Omicron. It was first detected in November of 2021. So that's this asterisk right there. And although you see all of those orange and red dots are the Omicron sequences of BA1 and BA2 and BA4 and BA5. But those dots don't start until around then. That's when we first detected it. But if you look back to their common ancestor, the common ancestor of BA1 and BA2 and BA4, BA5, that was 6 months earlier. And if you look at where in the evolutionary tree it last connected with the rest of SARS-CoV-2, that was a year and a half earlier. So where was it for a year and a half that we didn't detect it? And yet it was evolving. It was evolving somewhere. So where was it evolving that all of a sudden it would appear with so many genetic differences from the rest of the strains that we were sequencing? Well, there's smoking guns, and the smoking guns are all pointing to passage through immunocompromised individuals with chronic or persistent infections. The reason why we think this is true is that those we know of individuals that have been infected with SARS-CoV-2 for over a year. I'm not talking about long COVID here. I'm talking about a chronic infection. The virus is replicating within individuals and their immune system is not able to get rid of the virus. That can happen for a number of if you're immunocompromised or immunosuppressed for a number of reasons. If you have cancer, if you have active uncontrolled AIDS, if you have other immune disorders, you can have persistent COVID infections. And sometimes these people who are chronically infected don't even know it. I had a friend who was going in for cancer treatment and they couldn't get treated until they couldn't have the surgery until they cleared COVID. They didn't know they had had COVID. They had no idea that they were carrying the virus. So some of these infections are very long lasting. And in such individuals, every single one of those individuals, evolution is happening within their body for this virus. There can be high and prolonged viral replication. And every individual is going to have a slightly different immune environment, a slightly different cellular environment, and the virus is evolving in that patient to that individual, allowing mutations to accumulate, especially in the spike protein, where it's interacting most strongly with the host cells. And that evolution within a patient is hidden from surveillance. There are about 2 or 3% of people that are immunocompromised in various studies suggesting that these chronic infections could be pretty widespread. So a lot of these events can be happening at any point in time. And it helps explain where these hidden lineages were. They probably were in a patient or two. It could have transmitted from one to another, and that could have led to this divergence between the Omicron lineages. And it also helps to explain these kind of jumps. Those jumps weren't jumps. If we had gone in and understood what was happening in the patients in which they were evolving, we would better understand, oh, they were accumulating mutations for the virus to be able to better infect and live inside that patient. But once it hopped out, then we saw the changes for the first time as it spread through between people. So that's our best understanding of why these lineages these variants of concern have arisen with many unusual features, a suite of mutations and a deep evolutionary history, but no previous detection. They probably were passage through immunocompromised individuals. And here's just one example of the type of data that leads us to suspect this. These are genomic comparisons from a study by Harari et al. Of chronic infections. This is the genome, the 30,000 base pairs in the genome, and where mutations have been documented in a series of papers. And chronic infections tend to have more mutational changes in spike. That's in pink here, this the spike protein that interfaces with our body, with our host cells to get into our cells and also to evade our immune system. So that changes more rapidly in chronic infections. Also changes more rapidly in the variants of concern. These are the mutational unique changes that happen in each of the variants of concern. And again, a pattern of more spike mutations, where compared to just the kind of gradual evolutionary change that's seen across the genome in most of the global sequences. I'll just really quickly go through it. And we've also case studies of patients with chronic infections have also mimicked the types of changes that we've seen in variants of concern. This was one study from Cell, Host, and Microbe of a patient that had infection for 190 days, and they tracked they were trying to clear the patient of COVID, but they came back repeatedly and they sampled the virus over time and tracked it as it evolved and got changed over that time period. And this is a little bit of a complicated slide, but I only want to take one part take-home message from this. Over that 190-day infection, that virus within that patient evolved to be something like a Wuhan strain to something that by the end had the evasion properties much more like beta, the variant of concern beta. So they tracked within that individual the evolution of the virus in such a way that it mimicked one of the variants of concern that was circulating earlier in the pandemic. Not from this patient, but that just goes to show it was tracing a similar path in this patient. So this one really important take-home message from that is that we have to globally prioritize detection and treatment of those suffering from persistent infections. Inequities in access to medical treatment across the world are huge. Many people do not have access to treatment for HIV. It's extraordinarily expensive and out of reach for the vast majority of people that have HIV. And unless we treat the conditions that lead to immunocompromised status and look for and treat SARS-CoV-2 infections, then we should expect to see these type of evolutionary jumps again and again and again in the future. And when I, early on in the pandemic, when I was talking about the need for this on one of the public health calls, a doctor said, but what can we do about this? And I said, well, are you actually regularly testing your immunocompromised individuals just to see if they carry SARS-CoV-2? And they're like, oh, no, I guess we're not. And I think that's just an example of where evolution can interface with public health and slightly change the practice of medicine going forward. So I think with that, that's plenty to have shared with you, and I'm sure you have lots of questions. So let's stop there and take questions.
Amazing. Thank you, Dr. Otto. That was an amazing talk. So I'll turn the floor over to our students in the audience for questions. Please feel free to use the chat or raise your hand in Zoom and we'll take your question.
Shall I take those, Marcia, or do you want to?
Oh, yeah, if you'd like to, definitely. We can start with Tyler. I see their hands up, so.
Great.
Okay. Hi, Professor. Thank you for your speech. And I have a question for you about in the speak of evolution. I heard that there are saying that modern people have stopped from evolution and even a bit going backward in the sort of physical representation. I'm not quite sure is how much of that speak is true, and are we still really evolving?
Yeah, you can't stop evolution. Evolution will continually happen. You can, medicine is changing the selection acting on our genomes. So for example, I would have died in childbirth. Childbirth was frequently a cause of maternal and infant mortality until recently, but now with C-sections and ultrasounds, many of those childbirths aren't fatal anymore. So that's changing selection. For example, on infant size, that kind of mismatches between the pelvic girdle of the mother and the baby's head are no longer fatal in the way they would have been. So it's changing the selection, but mutations are still happening and selection is still happening.
Okay, thank you.
And there's some questions in the chat. I'm not sure, Sally, if you can see that or I can read them out.
Yeah, so I'll just start at the top.
Sure.
Jasmine asks, what biological processes are behind COVID's ability to evolve so quickly? And I think that there are two parts of the answer. First of all, it's an RNA virus, and RNA viruses in general aren't as RNA is more unstable, so it tends to accumulate mutations more readily. That said, it does have an error checking gene in it, so it's not even the most mutable virus. Flu is a more mutable virus than SARS-CoV-2. So it's not just that SARS-CoV-2 was spewing out or gathering mutations faster and faster and faster. But still, it's an RNA virus and it does accumulate, what did I say, 1 genetic change per genome every 2 weeks. I think the bigger issue is there's just so much of it. There's so much COVID globally. There's so many persistent infections globally that basically every single base pair change in the whole genome and multiples of them are appearing in some individual somewhere on the planet. And that just gives this virus an enormous ability to kind of track and change, kind of explore the different genetic variants that could be more favored, and then for those to spread. So I think that's the main reason. And then there's another reason, that is that because it's fairly new in humans, I think it's still adapting in ways to better infect us. So that's also probably going on as well. Okay, second question. Eric says, how do you know your variant data is representative? If it is mostly from hospitalizations, wouldn't that overrepresent more dangerous strains? That's a really good question, Eric. So the reason that when we had data from that was more representative, we didn't see a difference between hospitalized cases and outside of hospital. And we wouldn't expect that either for SARS-CoV-2, because when these variants are spreading, they spread through the community. And so rapidly, those strong selection coefficients mean that if XEC is spreading at 3% rate, it's spreading broadly through the community at that 3% rate. So we don't tend to see much of a difference between what is in the hospital and what is not. But you are right that when the variant spreads, one of the questions that we need to track is whether or not that variant lands people in the hospital more often or less often than before. And with Delta and with Alpha and with Beta, it was more deadly. And fortunately, with Omicron, it is less deadly, that particular variant. And that's just among uninfected, unvaccinated individuals. I can pull up the plot. I think I'll leave it at that and just say that that is a great question and one of the things that we try and tease apart. Could you talk more about your current recent research that you mentioned earlier about studying the limits of evolution? Yeah. So we are using heavy metals, copper, nickel, cadmium, and putting so much in the medium with that yeast grow in, that they slow down in their growth until eventually they can't grow anymore at all. And we repeat this hundreds of times, so most of them go extinct, but every once in a while we'll see growth in one of the wells of our experiments. And those wells that show kind of precipitate because that yeast is growing so much that they're clouding the medium and precipitating, that's telling us something's changed. So then we pull those out and we sequence and determine what has changed. And what we're trying to do right now is, well, humans aren't just changing the world in one way. We're not just causing temperatures to rise. We're also causing new invasive species, pollution, plastics, all sorts of different environmental changes. And one of the things we're looking at now is, can we predict at all which combinations of stressful, toxic conditions make it impossible? So we're putting metals together and finding out whether it's which combinations of metals make evolutionary adaptation. There's just no mutations that allow growth to happen. What are those limits? And can we generalize that to say anything about what predicts which combinations of environmental changes are just going to be too much? Questions. I'm loving these questions, so keep them coming.
The students always have great questions. So we'll give you a moment to type those in the chat, or feel free to raise your hand. Maybe while we're waiting, I'll ask a question. I have two, actually. One, I wondered if you could tell our students a little bit about what it was like early on, perhaps in the pandemic, you mentioned this like, sort of big collaborative network, CoVarNet, across Canada. What was it like to mobilize and get everyone working together?
Uh, um, really a strong point was how scientists like put aside everything else to help, I think, and that's from, geneticists, immunologists, cell molecular people, just everybody was trying to pitch in and help understand this virus evolving. And it's a new disease. And so there was so much we didn't understand, and the science was changing every day. So it was great to be a part of an effort. Like, it was terrible to be surrounded by this pandemic. And we really have to remember how scary it was when death rates were so much higher. We didn't have vaccines. We hadn't had, and we didn't really have any immunity. Plus, it was a more deadly disease at that time. So I did find that slide. So I'll just share it. Here is the hospitalization rates, the hazard ratios relative to the Wuhan strain of alpha, beta, gamma, and delta. So those were much more, almost twice as virulent, delta in particular, and then Omicron about half as virulent, even without any immunity. Yeah. So, but I also have to say, there was part of me that was so hopeful because all the scientists and all the public health people were kind of coming together to work together. And I actually don't feel as hopeful right now, but I hope my hope returns. And that's because, people are sick and tired of hearing about COVID. Sorry, everybody. They're sick and tired of masks. There's more anti-vaccine sentiment than there has ever been, and that's dangerous. And right now in BC, we have a teenager in hospital with H5N1 avian flu. And this is not the time to have an anti-vaccine and an anti-mask oriented population or popular movement. So that scares me. And also the data that I was hoping would become more and more open has kind of gotten more and more closed over time. Canada has a data problem. Health authorities in the province are different jurisdictions. The provinces are all in charge of health, and those are not combined together in the federal system. So there was nobody in Canada that during the pandemic, had access to the genomic sequences and knew which viruses were killing people and which viruses were not killing people, because that data was in the provinces and not shared. So they had the genome sequence, but the information linking it to what happened to that person was not shared. So that's a problem, and I think we need as a country to really solve this data problem so that we can answer questions like, here's a variant spreading and it is more deadly or not so deadly. Either way, we need to know.
Yeah, the information is critical, and I guess like kind of dovetails to my next question, and then we'll go to Larissa's got a question in the chat. But you mentioned that the variants of concern that are sort of making these leaps may be happening in immunocompromised patients. And so if that continues to happen, do you not see kind of an endemic kind of phase where we then can predict what's happening with SARS-CoV-2? Is it going to kind of persist in this same pandemic phase where it's less predictable?
So I think right now we're in the kind of endemic phase, by which I mean that there's waning, and that waning of immunity is allowing more cases to grow. So it's kind of at a steady state. It's not flat. And that's because we keep seeing these variants that kind of bubble up and then lead to a little peak, but then they run through everybody. And so the immunity rebounds and pushes it down again. So we're seeing these like little ebbs and flows, not these huge waves, but the variants that we've seen lately, for the last 6 months are all these BA.2.86 variants. And so we haven't seen another jump yet. But those jumps, I cannot stress enough, evolution is predictable in the short term. But what makes it unpredictable is we don't know what combinations of this virus can do a different thing. The shift from before Omicron to Omicron was, I don't think, very predictable. Omicron gets into our cells differently. It attacks different tissues. It tends to be more of an upper respiratory disease and not a lower lung disease. And that's why it's not quite as deadly. But that jump to a new way of making a life for the virus, that's what's hard to predict. And the more chances we give this virus to kind of make those big leaps, maybe the next big leap will be something that can grow well both in our upper airways and in our deep lungs, and that would be bad. More transmissible and more deadly. So that's what we don't know. We can't really predict what's outside of the immediate surroundings of what we see. There was a question about diploids. Larissa, yes, there's a similar equation for diploids. You can come and take our classes. Evolutionary fundamentals of evolution in 336 goes through the fundamental equations for diploids as well as haploids. And I teach a biomathematics class. You can take that in any year, but that kind of gives you more comfort. Like when I see those equations, they talk to me. I know what they're saying, and I can translate back and forth. But if you're not used to equations, they can look like a black box. And so really to understand a lot of these models and statistical analyses, you want to get fluent in the language of equations. So biomathematics 301, biology 301 is a class that I do to get familiar with building and analyzing those models. Correct me if I'm wrong, but how come COVID-19 did not affect animals like pets, even though there were so many different variants? Well, they did. So some animals were not that susceptible. So initially rodents weren't that susceptible. Before Omicron, they weren't. They're more susceptible to Omicron. But then even among animals that could get infected, some had terrible symptoms and died, and others were pretty asymptomatic. I don't know why, but cats tended to have been asymptomatic, but mink, not so. So the SARS-CoV-2 ran amok through mink farms, leading to massive mortality and ultimately killing of those mink to prevent it from further spread. So there have definitely been pandemics within animals as well. And in wild populations, white-tailed deer, for some reason, tend to be a reservoir now. It's from humans, it's escape into white-tailed deer, and white-tailed deer tend to have a high prevalence of SARS-CoV-2. So I think that is an open question, Larissa. Was it Larissa? No, it's Rana. Just how many animals are hosting or suffering from SARS-CoV-2 as well? Okay, can you go deeper into how predicting evolution works? So I mentioned that we have equations, and once we know the selection coefficient and the current frequency of an allele, I can predict how frequent it will be 1 day in the future, 10 days in the future, 3 months in the future. And so those were those straight lines on this logit plot that I mentioned. And so that's very predictable. But I can't predict what's going to happen in a whole year from now. And that's because there will be new variants that emerge. So that's the mutational process is less predictable. What new mutations will appear, and will there be any of these kind of big jumps? There will be, but I don't know what they are. What will these next big jumps be, and what will they do? So that's the unpredictable part. So Arjun is saying, you talked about how the Omicron variant was the least predictable in terms of its rate of spread in the beginning. Well, before it appeared, we just didn't know, again, when the variants of concern arise, it's a real moment of uncertainty among all scientists because we detect this very, very different variant. And at first, we don't know how fast it's spreading, and we don't know how deadly it is. So these big evolutionary jumps with many, many mutations are extremely hard to characterize until we do the lab work that says how quickly it gets into our cells. We do the lab work that tests how well does our immunity to this virus hold up against the new variant, and over time detect whether or not people land in hospital more or less often. But at first, there's all this uncertainty when a new big jump occurs. Okay. Would it be okay to say that it may have been due to people opting to do, okay, so then it's about Omicron in particular, opting to do rapid tests at home, making the data less available, or the variant causing less severe symptoms, meaning less hospitalizations, and again, leading to less data being available about the variant to plot on. So by the time Omicron was spreading, those, so it was unpredictable when it would occur in a province, but once it was there, it was very predictable how fast it's would rise in frequency. So it was really those early days before it was even in the country that it was less predictable about when would it get here and what would the consequences be. But we learned from other countries. We learned from South Africa. Their science, their genomics on SARS-CoV-2 has been excellent. And we've learned a lot about the features of these viruses before they even entered into Canada. So it is, I'm a big fan of at-home rapid tests. If you've got any symptoms at all, test. Stay home if you've got COVID. And that just reduces the burden of disease on others around you. And what we saw with Omicron was, remember those really long lines to get tested? We didn't have rapid antigen tests before that time, but they just started really rolling them out. You can get them at the pharmacy and people stopped testing. So it was harder to know how many cases we had at that time, but it was, despite that, even though we didn't know how many cases there were, the frequency of Omicron within the population was still obeying these S-shaped curves, evolutionary curves. So the models could handle that, the change in detection. And then can you briefly describe how air filtration affects the spread of COVID? Yeah, so it's a spread in the air, spread it through aerosols. And so anything we can do to increase ventilation in rooms is great. One way to reduce the spread of COVID from infected individuals to those around them. Open the windows. Right now, as I said, we're predicting a rise in cases with XEC spreading. So open windows. I wish we, in some countries, they made mandatory carbon dioxide tests, kind of monitors in public areas, restaurants and other places. And I like that idea because it was telling you just how much air circulation there was. When we breathe out CO2, the CO2 levels in an enclosed space rise. And not just CO2 levels rise, but any virus we're breathing out would also be rising. So it's a good clue to being in a space that's not well ventilated. All right.
Excellent. Well, maybe with that, we will close there. Great questions from the students. And thanks so much again, Sally, for such an excellent seminar. I learned a lot.
Absolutely. Thank you all.
Thanks, everyone.
Bye. See you in a couple years.
Yeah, I definitely take Dr. Otto's class. Highly recommend it.